Okay, how do we determine the gender in a human being? Of course, I already briefly mentioned we have 22 pair of autosomes, meaning that we have 22 pair of chromosomes that determine our body phenotype, the phenotype in our body. And then the 23rd pair, we call them sex chromosomes. For male, it is XY. For female, it is XX. So the term X and Y, they are the name of the chromosome, not the gene or the allele of the chromosome, okay? We have an X chromosome, we have a Y chromosome, okay? So for the 23rd pair, for female, you have 2 X chromosomes. For male, you have 1 X chromosome and 1 Y chromosome. And Y chromosome is very short. So you can assume, okay, in our level, since this is introduction to biology, you can assume, okay, assume Y chromosome carry no gene, okay? You may call it genetically empty. We can assume that you will make your understanding of the Punnett square easier later. Genetically empty. So you may assume that Y chromosome does not carry any gene. So the gene is only carried by X chromosome. That will make your life easier. Y chromosome in reality, of course, Y chromosome carry genes, okay? If you take my physiology, I will tell you there's one example of the Y chromosome gene. It's called SRY gene. Sex determining region on the Y chromosome. This is called SRY gene. But it is beyond our discussion in here. You can just assume that Y chromosome has no gene. So when the X and Y chromosome, I would say when meiosis occur with error, what happens is that you will cause sex chromosome disorder. The term non-disjunction, meaning that the homologous pair of the chromosome, they do not separate. They go into one cell. So the homologous pair, they do not separate, and then they go into. So one cell will have the homologous pair of the chromosome, the sex chromosome. Specifically, we have the sex chromosome. The other cell will have no sex chromosome. And then during meiosis 2, you will have you separate the chromatids, okay, and then fertilization occur. Then some of the zygote will have the free sex chromosome. Free sex chromosome we call it trisomy. After fertilization, the cell only has the chromosome from the sperm. Of course, we call it monosomy. You may or may not have heard about this, but the most famous trisomy disorder. Okay, trisomy disorder here is not just sex chromosome, it can be your autosome, the body chromosome. The famous one that occur in your body chromosome is the Down syndrome. And it is a trisomy at the chromosome number 21. Chromosome 21. It is not the sex chromosome. If you have trisomy in the sex chromosome, we have other sex chromosome disorder. For the body chromosome, for example, chromosome number 21, if you have trisomy, that means free chromosome in the body chromosome number 21, then you will have Down syndrome. So previous slide shows you that there's a non-disjunction in the meiosis 1. What if you have non-disjunction in meiosis 2? Non-disjunction in meiosis 2 greatly decreases the chances of trisomy. You only have 1/4 of the chance to have trisomy and 1/4 of the chance to have monosomy. Remember in the previous slides, if you have a non-disjunction, that means the homologous chromosome, they do not separate in the meiosis 1, then you have 50% of the chance to have trisomy and 50% of the chance to have monosomy. But in here, you have 25% of the chance you get trisomy and 25% of the chance you get monosomy. The other 50% of the chance you will have normal, healthy diploid. So here is the summary of the chromosome disorder. So as I mentioned before, the famous one is the Down syndrome. It can occur in either male or female. It is because of trisomy 21. The chromosome number 21, which is an autosome, you have 3 chromosomes in chromosome 21. So the total chromosome number is 47 instead of 46. And then Poly X, Klinefelter, Jacobs, and Turner. All these are sex chromosome disorder. I'll just briefly talk about this. Poly X meaning that it occurs in female who has triple X. Well, it's like the movie Triple X. Or 3 X chromosome. Okay. So for triple X female, they are tall and skinny. They may have delayed motor or language development, but they have a normal mental capacity. They have normal mental capacity. So there's not much of the it doesn't affect female too much. But Klinefelter occur in male because it has the patient has Y chromosome, but the patient has 1 extra X chromosome. That causes the male develop some female feature. For example, the male can develop a breast. So other than developing breast, they will also have difficulty to reproduce. So they may need artificial help, artificial insemination, or some kind of intervention to help them reproduce. And they may also have delay in the speech development as well for the patient with Klinefelter syndrome. For Jacobs syndrome, it also occurs in male. They have extra Y chromosome. They are very normal. They are pretty much normal male, except that they may have acne problem on the face and on the body. It doesn't affect their reproduction. It doesn't affect their physical body, except maybe with excess acne. Okay, last but not least, the Turner syndrome. Turner syndrome occur in female. They only have 1 X chromosome. So this is the monosomy. What happened to those female? They are usually shorter than average, and they do not have. They have normal range of intelligence. They also develop a slower speech acquisition, and also the interesting thing is that their reproduction is also impeded. So they may not have a menstrual cycle. The ovary may not may even degenerate or may not even give out eggs. So what is the treatment? Is the hormone replacement therapy. Usually people give the Turner, a female patient with Turner syndrome, usually doctor prescribe a female hormone to them. Okay, let's talk about chromosome mutation. Previously, I talked about the gene mutation. We talk about gene mutation. Usually, they do not produce obvious phenotype. Most of the gene mutation is neutral. That means there's no effect or no changes. But chromosome mutation is different. Chromosome mutation usually will involve several genes. Because it involves several genes, then usually we'll see some kind of effect or changes in the phenotype. What causes chromosome mutation? Radiation, organic chemicals, or viruses. Those can cause a chromosome mutation. Or chromosome mutation is caused by chromosome break. So chromosome can break in different ways. So these are the 4 different ways that we'll talk about. Breaking of chromosome: deletion, duplication, translocation, and inversion. These are the 4 ways that can cause chromosome mutation. Okay, 4 ways to cause chromosome mutation. And this kind of chromosome mutation can occur during meiosis. Okay, let's take a look at each case. Deletion is very simple. So the gene H here, after the meiosis, it comes out. It just disappears. It is lost. So part of the chromosome is lost. So the entire chromosome becomes shorter. That's why we call it chromosome break. And then translocation. Translocation is that the gene, for example, GH and the gene QR, they exchange the location. So originally gene QR is on the shorter chromosome. Now it goes to the longer chromosome. Gene G and H originally is on the longer chromosome. Now it goes to the shorter chromosome. You may ask, hey, isn't that the same as crossover or we call it recombination during prophase 1? No. Remember crossover or recombination during prophase 1? The gene jump across a chromosome within the homologous pair, homologous chromosome. That means some of the gene from your father chromosome goes to your mother chromosome, and vice versa. Some of the gene from your mother chromosome go to your father chromosome. So they are a pair. They are the same chromosome, same type of chromosome. But the translocation that we talk about here, it occurs in different chromosomes. For example, okay, I just randomly assign the number. Maybe this is chromosome number, the longer one may be chromosome number 1. The shorter one may be chromosome number 20. In other words, the gene, they are moving to different chromosome. That is called translocation. If the gene move from chromosome number 1 to chromosome number 1, that means if the gene, they are moving within the homologous pair, then we call it crossover, which occur commonly in prophase 1. That is normal. But if the gene move across chromosome, let's say from chromosome number 1 to chromosome number 20, they are different chromosomes. They are not homologous pair. Then this is called chromosome mutation. We call it translocation. Okay, inversion. What is inversion? inversion occurs in the homologous pair. So you can see that, okay, these two are the homologous pair, but the gene here, they are inverted, because at this locus you should supposed to have a C gene here, but this chromosome you have the E gene, and this locus you should have the E gene, but now you have the C gene. So that's why we call it inverted. Okay, and then crossover occur. When crossover occur, it gets even messier. You can see that, okay, you can see that here it should be the AB gene, but AB gene is gone. Theoretically it should be the AB gene in here, but the AB gene is gone. Oh, it should be small a, small b here. Therefore, we call this deletion, okay? And then the AB gene, it becomes here. So the AB gene is here, and they do not pair with the original EFG here, because it is inverted. It is inverted, and it. So last one, duplication. Duplication is very simple. So you have the D and E gene here. You also have D and E gene here, but somehow during the replication, during the meiosis, you replicate E and D gene, and at the same time they are inverted. So the entire chromosome increase the length. So just remember that we have deletion, translocation, duplication, and inversion. 4 types of chromosome mutation.